Welcome to LesionTracker. If you are a new user, please click Need an Account. For registered users, enter your email and password and press Enter on the keyboard or click Sign In. After login, you will be directed to the study list page. This page is sortable and filterable by each column heading. For example, I can sort by medical record number, or study date, or filter by patient name. To make a time point assessment, identify the corresponding imaging studies that you would like to associate. Hold down the Shift key on your keyboard to select multiple studies. Then right-click with your mouse and select Associate. Study List Association will display all imaging studies within 14 days of your study list selections, just in case you forgot to include a related study. From this modal, you can select or unselect studies to include in the time point assessment. And identify if the time point is baseline or follow-up. Click Save to save your association. Double-click a study to view the associated time point images. The Series and Study Quick Switch will allow you to easily change between series and studies. Alternatively, you can use the Study Select panel to switch between studies or series, or select a different time point if available. The LesionTracker has standard tools to window level, pan, zoom, and scroll. To window level, use the keyboard shortcuts on the number pad. Or you can click the window level icon on the toolbar and then press and hold the left mouse button to manually change the window level. To zoom, press and hold the right mouse button. After selecting Pan from the toolbar, press and hold the left mouse button to move the image around the screen. To scroll through the images, there are three options. You can use your mouse wheel, the scroll bar, or the up and down buttons on your keyboard. LesionTracker supports conformance checks for RESIS 1.1. Click Trial to view response criteria details. These checks are applied for baseline time points. Click Close to dismiss. LesionTracker provides tools to create target measurements and non-target annotations. To create a target, select the target icon from the toolbar. Then click on the image, hold, drag, and release to create the target. To label the target, click Add Label. To edit the measurement, just drag the endpoints. If you'd like to rename or delete the lesion, click on the lesion number in the table and then click Rename. To delete, click Delete. This will delete the lesion from the table and the image. To create a non-target, select the non-target icon from the toolbar. Again, click, hold, drag, and then release to create a non-target. Select the label from the list and confirm the response. LesionTracker will flag any lesion that conflicts with the response criteria conformance checks. To review why the lesion has been flagged, click the icon to the left of the lesion record in the measurement table. This lesion was flagged because 3 lymph nodes were taken as target, and RESIST only allows 2 targets per organ. This target was also flagged because it does not meet size criteria for lymph nodes according to RESIST 1.1. When you are finished identifying all targets and non-targets for the time point, click Generate Report at the bottom of the measurement table. This will create a PDF which includes all targets and non-targets and associated annotated image snapshots. To exit, click Save or Study List to go back to the study list. To compare images for baseline and follow-up time points, first identify the related imaging studies for follow-up, right-click, and select Associate, as shown earlier in the video for the baseline. After follow-up association, double-click a follow-up imaging study to launch into compare mode. Again, use the Studies panel or the Series and Studies quick switch to select the series you'd like to use for measurement. The first target is displayed by default for the baseline study. Scroll through the images for follow-up to find target 1. Select Target tool from the icon and make the measurement. Targets and non-targets must be created in the same order as they were on baseline. To advance to the next target for baseline, click the lesion in the measurement table. So clicking on target 2 will show target 2 for the baseline measurement. Scroll through the images on follow-up to find target 2. In this case, the lesion has resolved. Select the CR target tool to denote a completely resolved lesion. In the case when a lesion is not imaged or can't be evaluated, use the Unknown Target tool. Continue adding targets and non-targets to the follow-up until all disease has been assessed. This icon will display assessment progress. 2 out of the 6 lesions, including targets and non-targets, have been assessed. This displays the total targets that are measured on baseline. Once all target measurements and non-target annotations have been made for the follow-up time point, it's time to review to confirm that nothing was missed. LesionTracker has tools to make image review more efficient. To easily compare lesions to ensure that they have been consistently assessed, click on the lesion record in the measurement table. To scroll through both time points simultaneously, activate the Synchronize Scrolling option via the More menu on the toolbar. If you wanted to check the size of a lesion without adding it to the table, use the Temp tool. There are additional image display and manipulation tools available in the More menu on the toolbar, such as Rotate, Flip, and Magnify. You can also increase the size of the image viewports by hiding the measurement table. LesionTracker offers a filterable audit trail. To view the audit trail, click the configuration icon and select View Audit Log. There are options to filter by user, date range, and action. Finally, LesionTracker also allows users to modify the color scheme to change the look and feel of the application. From the configuration menu, select Themes. LesionTracker offers 7 different themes, including our default theme, Tide. Select the new theme of your choice and click Apply.